Hello everyone, this is the first International Conference on Chemical Matters and Environment Preservation. This presentation is entitled Eradication Bacteria from Antibiotics to Metal Organic Frameworks or MOFs. It will be presented by me, GHANIA Oral Gha Nia, a PhD student at University of Ouargla. In this presentation, we will take some headlines as mentioned in the following plan. To start with introduction, what is an antibiotic? How do antibiotics act to eliminate bacteria? What are the different mechanisms used by bacteria to gain resistance to antibiotics? Metal organic frameworks, the antimicrobial mechanism of MOFs, and finally, conclusion. The growing resistance of pathogens to conventional antibiotics has become a public health problem and raises the need to seek new effective solutions. Metal organic frameworks are porous hybrid materials comprising metal ions linked by organic binding ligands. The possibility of using a variety of chemical building components in MOFs enables the formation of structures with desired properties. Let's begin with a definition of an antibiotics. Here are three definitions. The first one is according Selman-Waxman, who defines antibiotic as a chemical substance of microbial origin that possesses antibiotics powers. The second definition is according Webster's Third International Dictionary, a substance produced by a microorganism as a bacterium or a fungus and in dilute solution having the capacity to inhibit the growth of or kill another microorganism. The third one is according Brock's Textbook of Microbiology, a chemical agent produced by one organism that is harmful to the other organisms. The antibiotic has several mechanisms that it uses in order to inhibit and kill bacteria. Here are some of them. Inhibiting the synthesis of a bacterial cell wall, inhibiting the synthesis of proteins, inhibiting the synthesis of DNA, inhibiting the synthesis of ribonucleic acid, and finally a membrane disorganizing agent. In the same way that antibiotics have their mechanism to kill bacteria, even bacteria has its mechanism to avoid the effects of antibiotic on it. The first mechanism is acquisition of alternative metabolic pathways to those inhibited by the drug. Second one is decreased permeability of the bacterial cell wall, which restricts antimicrobial access to target sites, overproduction of the target enzyme, and degradation of the antibiotic, and lastly, modification of antibiotic targets. Now let's talk about MOFs as an alternative of antibiotic and how they act against bacteria. A MOF is a coordination polymer with an open framework containing potential voids. In turn, a coordination polymer is defined as coordination compound continuously extending in 1, 2, or 3 dimensions through coordination bonds, where the building blocks of MOFs can be held together by several types of bonding, and this diversity can be present in a single MOF crystal. Such bonding types include metal coordination, hydrogen bonding, electrostatic interactions, and pp stacking. This is one of the reasons why the structure of a MOF can be easily tailored to achieve characteristics required for a chosen application. MOFs can be synthesized through a variety of methods, from conventional, solvothermal, hydrothermal synthesis, and many other methods. This picture shows different structures of MOFs. Here are the antibacterial mechanism of MOFs in a nutshell. As a result of their many benefits, MOFs appear to be excellent candidates for biomedical applications. The antimicrobial activity of MOFs can result from the gases incorporated in the particle voids or properties of the metal component in the framework. Given that the antibacterial action of MOFs is associated with physical damage to bacterial cells, which occur via several mechanisms, MOFs are promising supports of or alternatives to antibiotics. This picture summarizing the most important mechanism of MOFs in order to kill or inhibit bacteria. where MOFs are working on inhibiting of the cell metabolism by releasing the ions that were gassed in their pores, where they was released in or out of the cell. These ions affect the bacterial cell via several strategies as is shown in this picture. For instance, these ions could cause DNA damaging and affect correct function of plasma membrane proteins. In addition, disruption of cell wall and plasma membrane components. Furthermore, they cause disruption DNA replication and enzymes, also decreasing the ATP level and affect reactive oxygen species generation. All these different mechanisms do the trick to inhibit or kill bacteria. In conclusion, the antibacterial mechanism of action of MOFs, which described in different studies, depends on the type of metal in the structure. The application of metals as an antibacterial agent is well known, but the particular advantage of MOFs is their ability to act as a reservoir of metal ions and to release those ions gradually. Here is the end of our presentation. Thank you for your attention.